Welcome back to the Cambridge Neuroscience Interdisciplinary Seminar Series. This series features current work from the neuroscientists across the schools and departments of our university, reflecting the pioneering work and diverse interests of our members here. Each seminar focuses on one of the new interdisciplinary themes of Cambridge Neuroscience, which we will be launching later this year. For more info on the talks covered in this seminar series and all things neuro-related in Cambridge, please follow us on Twitter at CamNeuro and follow the links below.
So I'd like to welcome everyone to the Cambridge Neuroscience Interdisciplinary Seminar Series. And today we're really delighted to welcome Professor Sir Simon Baron-Cohen. Simon is the director of the Autism Research Centre here in Cambridge. He is a joint professor at both departments of psychiatry and psychology, and he's also a fellow of Trinity College. So I'm sure most of you will know Simon's work. He's published many books on his subject area of interest of autism, but also published in excess of 600 papers. He has many kind of notoriety, like fellows of many different organizations, and they're all listed on the details for this talk. So we're really excited to welcome Simon here today, and he's going to talk to us on the prenatal sex steroid theory of autism. Welcome, Simon.
Thank you, Dervila. Thank you for inviting me to talk in this series. So you've already announced my title, Prenatal Sex Steroid Theory of Autism. This is the overview that basically I'm going to be talking about autism, and I'll explain what that is for those of you who are less familiar with it. And it's been quite well established now for over 2 decades that there's a genetic factor in the etiology of autism. But what our group has added to this story is the prenatal sex hormones factor. So this isn't a theory about hormones working in isolation. This is a theory about how these prenatal sex steroid hormones like testosterone and estrogen interact with a genetic predisposition to change prenatal brain development, and how this might affect boys and girls differently. So that's the kind of overview, and then I just kind of lay it out for you. And then at the end, we'll have time for questions and comments. But first of all, what is autism? So most of you know that it's diagnosed on the basis of social and communication difficulties. It's known as a neurodevelopmental disorder or condition. The second feature is about difficulties adjusting to unexpected change. So these are kids who get very stressed if things are not predictable. And they like repetition, they like predictability. Another feature in the diagnosis is unusually narrow interests, sometimes called obsessions, but some people might think that's a bit stigmatizing. They simply get quite passionate about particular topics. And then in 2013, the DSM, Diagnostic and Statistical Manual, from the American Psychiatric Association added sensory hypersensitivity. So that's kind of how the diagnosis is made. But alongside that kind of core definition of autism, most autistic people have co-occurring conditions. And this is a partial list. So a proportion of them have epilepsy or learning difficulties, so below average IQ. Some of them are minimally verbal. Some of them have other medical conditions like gastrointestinal pain or other neurodevelopmental conditions like dyspraxia, dyslexia, language delay. And sadly, many of them develop poor mental health. You can see at the bottom of that list. So when we're thinking about autism, we have to separate the kind of core diagnostic features from these co-occurring conditions. And what this tells us, just whilst we're on the kind of overview, is that autism is now very common. So this is data from the Centre for Disease Control in the US, suggesting that the prevalence of autism is 2.3% of the population amongst 8-year-old children. And that equates to 1 in 44. But the graph also shows you that the prevalence of autism has been increasing over the years. And probably this just reflects better recognition, greater awareness, and also that we have broadened the category, the concept of autism, particularly to include autistic people who do not have learning difficulties. So when I started in this field, now almost 40 years ago, we thought that autism was very rare. And all the textbooks back in the 70s and 80s said it was 4 in 10,000. Today you can see it's very common, 2%. But back then, we tended to focus on a narrow slice of what we think of as the autism spectrum today, which was that most autistic kids had learning disabilities. Whereas today, the kind of profile epidemiologically has flipped over entirely. Now the majority of autistic people do not have learning disabilities. So we've got better at kind of making the diagnosis accessible to people who are in mainstream education, for example. So relevant to the talk today, most of you know that autism is diagnosed more often in boys and males than in girls. So this is a graph from 2019. And what you can see in blue is when boys get their diagnosis and what the incidence is. And then in black, when girls get their diagnosis. This is over childhood and adolescence. And what you see is that the incidence is higher in males. So it's currently running at about 3 males for every 1 female get a diagnosis, but that females, when they get their diagnosis, tend to get it later. And there are various kind of speculations as to why that might be. One idea being that there is greater stigma about autism in females because our culture and many cultures places a sort of a greater expectation on girls to be social and communicative. And so girls who might have undiagnosed autism may be hiding it. In the literature, it's called camouflaging, until it becomes too difficult to hide their autism and they seek a diagnosis, particularly in secondary school. But you can see there is a sort of still quite a significant sex ratio bias towards males. And part of the reason why we started looking at the prenatal sex steroid hormones like testosterone, which are produced at higher levels prenatally by the male fetus, was precisely to try and understand this biased sex ratio. So let's have a look at autistic traits, because that's another concept that's important to understand. So far, we've just talked about autism as a categorical diagnosis, but you can also look at individual differences in terms of autistic traits. So just ignore diagnosis. And our group developed the AQ, the Autism Spectrum Quotient, now 20 years ago, which is a measure of autistic traits in the population. And in this 2018 study, we had the opportunity to administer a short version of this questionnaire, self-report questionnaire, to a large population, 600,000 people, online, obviously. But we were also fortunate that a very large number of autistic people took part, 36,000 autistic people. This was mostly in the UK. And I think it may be one of the largest studies of autism, certainly at that time, But what we see is that, first of all, autistic traits are pretty much normally distributed in the general population. Slightly skewed here, but you can see we all have some autistic traits. So it's a matter of degree. Within the non-autistic population, we observe a sex difference. So males, typical males in purple, score slightly but statistically significantly higher than typical females in blue. And then autistic males and females are just shifted over to the right. So they are showing a kind of extreme of the typical male profile. But there is a small but important sex difference. Sorry, the slides are jumping around. There it is, in the general population. In terms of genetics, just to get that out of the way, where are we up to in the field? Well, over 100 genes have been associated with autism. These are rare genetic variants. So rare in the sense that they occur in less than 1 in 1,000 people in the general population, but occur more often in autistic people. These are genes that are expressed in the brain. But what you can see there as a caveat is that these rare genetic variants, mutations, for example, are actually only seen in about 5 to 10% of autistic people. Yet we know that autism is strongly genetic, it runs in families. So rare genetic variants are not yet able to explain the majority of autistic people. The list of genes here that you see comes from a website called Safari-Gene, which is a useful resource because every time there's a new genetic finding in association with autism, it's published on this website. So the list is growing. And I guess the other take-home message here is that autism is polygenic. It's not a single gene. Many of those genes that we just looked at show higher expression levels prenatally in the brain, which is kind of one of the pieces of evidence for thinking about autism starting prenatally. So the other part of the genome that's relevant to look at are common genetic variants. So we've already looked at rare genetic variants. Here we're looking at common genetic variants, and many of you will recognize that this is what's called a Manhattan plot. So when you do genome-wide association studies, you get this kind of graph. It resembles the skyline of Manhattan, where you've got the human chromosomes along the x-axis. And what you notice here is this red line, which sets the probability threshold for saying that if a common genetic variant, so a single nucleotide polymorphism, alleles that we all carry, is statistically associated with autism. And here you can see that in this very large study, 18,000 cases of autism, 28,000 controls, there were just 5 common genetic variants that cross the red line, meaning that they are statistically associated with autism. And there's a kind of point here, which is that you need large samples to detect these common genetic variants, because each of them may have only a very small effect in contributing towards autism. So you need big data to reveal that. And work is ongoing to expand sample sizes to even more than this, because we suspect that there are many more common genetic variants to be discovered. But we know that autism is not the whole story, because you can have twins, identical twins like these two sisters, where one is autistic and one is not. So if autism was 100% genetic, then monozygotic twins where one is autistic should show high concordance, both of them also, but the other one also being autistic. The fact that you can get these discordant twin pairs suggests there may be some non-genetic factors that interact with genetic predisposition. And in this study, what was found was differences in the discordant twin pairs one autistic, one not, identical twin pairs, differences in expression levels of some of the genes listed on the right. So this is kind of revealing a role for epigenetics, or factors that can operate on the gene to change gene expression levels, which in turn could affect brain development. So let's kind of transition to looking at the link between autism and typical sex differences in order to understand the role that prenatal sex steroid hormones may play. And we'll start off at the level of the brain. So what we see here is, well, two graphs. On the right is data from the UK Biobank showing that there are indeed typical sex differences, in this case in total brain volume between males and females, and also separately in grey matter and white matter. I can see Mike Lombardo is in the audience today, and he was a co-author on that paper. But on the left, what we see is data showing differences between autism and typical children across childhood in terms of total brain volume. In blue are the typical kids, in red are the autistic kids. So what it looks like is that on average, autistic kids' brains are larger or growing faster in these longitudinal studies, but that this group difference between autism and controls is hiding a sex difference that occurs in the general population. So one way of looking at this is that the autistic brain is not only growing faster, but also it is an extreme of the typical male pattern of brain development. In a study that's now almost 10 years old, Meng Chuan Lai, who's a PhD student and a postdoc in Cambridge, tested the relationship between autism and typical sex differences using VBM, volumetric-based morphometry. So what he did, in I think a very interesting analysis, is he first identified which bits of the brain show sexual dimorphism. So which bits differ in grey and white matter between typical males and females. Then he asked which parts of the brain differ between autistic people and non-autistic people, and then did an overlap analysis to see whether the differences between cases of autism and controls overlapped with the brain regions that show typical sexual dimorphism. And what you see is that particularly in females, the red lines there was a significant overlap. So the areas of the brain that are different in autism are significantly overlapping with areas of the brain which are different between males and females, particularly in white matter, but significantly also in grey matter. So one interpretation of this is that autistic females' brain is masculinized. In terms of differences in the brain, another way to look at the autistic brain is at post-mortem. And this data comes from 2014. And what we're looking at is individual neurons on the right from an autistic brain, on the left from a typical brain. And you can see using the naked eye that the little white dots that are along the length of the neuron are these dendritic spines, or where the neuron forms synapses with its neighboring neurons, that there are more of these in the autistic brain than the typical brain. So one interpretation is that the autistic brain is not only larger, and we'll see evidence from a study by Korschen and colleagues that there are more neurons in the autistic brain at postmortem, but also more connections between neurons in the autistic brain. And if we now sort of try to compare number of neurons in a typical female brain shown in green, male brain shown in blue, and then an autistic brain shown in red, we see that there are average differences between females and males. Males on average have about 22 billion neurons. Females on average have about 19, is it? Billion neurons. And autistic people, from evidence from Corchen's lab, have 65% more neurons, particularly in the frontal lobe, than typical males and females. So this is all post-mortem study. But again, one interpretation of what's going on is that the autistic brain may be an extreme of the typical male brain. Dori Flores, who was a PhD student in Cambridge, but has now moved on, published in 2018 some data using resting state, and particularly looked at the default mode network in autistic people compared to controls. And her work found a shift towards maleness in autistic people compared to controls. So we're seeing that when you have the opportunity to compare autistic people with typical males and females on many different metrics, there is evidence of masculinization, if you like. What about at the cognitive level? So this is now old data, but we've looked at one aspect of cognition, which is empathy, the ability to put yourself in someone else's shoes, to imagine their thoughts and feelings, and particularly used the eyes test, where you look at photographs of people's eyes and you have to say which of these 4 words best describes what the person in the photo is thinking or feeling. In big data, you can see published by Varun Warrior, who's a postdoc in Cambridge, women on average score higher than men on this empathy test. So 44,000 women took the test, 43,000 men. So it's big data showing a small but statistically significant difference in empathy. And when you get the opportunity to look at autistic people, as we did in this 2015 study, again, you see the typical sex difference, women on average having better empathy than men, and autistic males and females scoring below average, but once again, potentially interpretable as autistic people showing an extreme of the typical male profile. And this particularly old study using fMRI whilst participants are looking at the eyes and are having a functional MRI scan shows a sex difference in the left inferior frontal gyrus, whilst typical individuals are decoding facial expressions to infer what someone is thinking or feeling, and autistic people showing reduced activity in that same brain region whilst they're doing the task. So at a functional level and cognitive level, we may be seeing autism as an extreme of the typical male brain. Using a non-social cognitive task, this is the embedded figures test. You have to find this target shape as quickly as you can in the overall design. And decades of research have found that typical males are faster than typical females on this test. So this data shows you the average number of seconds, and in brackets, the standard deviation to find the target. I'll trace it out for you whilst we're talking. And typical females are taking slightly longer than typical males. Autistic people are super fast and super accurate on this test, reminding us that autism isn't just about disability, it's also about strengths and sometimes talents. And when you ask people to do that task in the scanner, you find that typical males, show less activity in the posterior parietal cortex whilst they're doing the task, and autistic people show even less activity in that same region. So both at a cognitive and at a functional imaging level, we're seeing patterns in the data suggesting that autism might be an extreme of the typical male brain. In terms of empathy, again, you can see we've got a different questionnaire which we used in that large population, the empathy quotient. So once again, 600,000 typical people, 36,000 autistic people. This time, typical females are scoring higher than typical males in the population on average. Those two words are really key, on average, because obviously these findings don't apply to all males and all females. And autistic people, males and females, are scoring below average on the self-report measure of empathy. And one other aspect of cognition that we've been interested in is systemizing. How interested are you in systems of one kind or another? Systems are defined as anything that follows rules. Could be a mechanical system, could be a mathematical system or a musical system, but where there are patterns or rules to be identified to understand how the system works. What we see here is that typical males report stronger interest than typical females in systemizing, and autistic males and females score even higher than typical individuals, but again suggesting that their profile on these dimensions of cognition, empathy and systemizing, might be an extreme of the typical male profile. And if we put those questionnaire data together, so we're plotting the empathy scores along the y-axis and the systemizing along the x-axis. With that very big data, just eyeballing the data, you can see in yellow are typical females. They tend to cluster up here. We call this type E, where their empathy is at a higher level than their systemizing. The green ones are typical males in the population. They seem to cluster more down here. And we call this particular profile type S, where their systemizing is at a higher level than their empathy. And then autistic people are shown in purple and pink down here, who seem to cluster more in type S or extreme type S, where their systemizing is at least intact or if not above average, but their empathy is below average. And that's just eyeballing the data. You can actually do a count to see how many people fall into these different brain types, as we call them. So you see more females in the general population show the profile of type E, where their empathy is higher than their systemizing. More males show the opposite profile. And then most autistic people seem to have a brain of type S or extreme type S, where they're kind of leaning more towards the world of patterns and systems rather than people. And one other way to reveal this is to calculate what's called a D score, which is a difference between your empathy and your systemizing. And what you see is a typical sex difference, and autistic people shifted over to the right with a higher D score, a higher difference between their empathy and their systemizing in a masculinized direction. There's an interesting sort of side story to this, which is in that big population, we could divide people who work in STEM or don't work in STEM. Obviously, people who work in STEM, science, technology, engineering, or maths, would be expected to have a strong interest in systems or be good at systemizing. And what we see is that people who work in STEM, on average, also have more autistic traits. So this is showing an interesting connection between autistic traits and aptitude in understanding patterns or systems. And I think this is an important finding in terms of taking autism outside of the clinic, looking at autistic traits in all of us and seeing what autistic traits confer, which seems to be an aptitude in understanding systems. And to test this at a molecular level, we had the opportunity to work with the company 23andMe. So many of you will have heard of it. It's a personal genomics company. So people give a saliva sample and get results to do with common genetic variants. And Varun Warrior and colleagues here in Cambridge were able to look at whether there was any correlation between the common genetic variants that are associated with good systemizing and the common genetic variants associated with autism, finding there was indeed a significant correlation. So again, this is changing our view of autism genetics away from the old medical model of looking for, let's call them diseased genes or mutations, and looking more at how combinations of common genetic variants in the population, particularly associated with aptitude in understanding patterns and systems, also predispose to autism. A lot of the sex differences that I've described to you so far, behaviorally, cognitively, have been reported only in adults or later childhood. So there's always that possibility that all we're seeing is the result of socialization or culture. Now, 20 years ago, we had some master's students who looked to see if there was a sex difference at birth before culture has had any opportunity to shape the human brain. So here in Cambridge at the Rosie Maternity Hospital, they presented babies 24 hours old with a social stimulus, the face, or a non-social stimulus, a geometric design, to see whether babies look for longer at one or the other. And the data that's being displayed here are those babies at 1 day old who looked longer at the face or longer at the geometric design. And what you see is that more girls than boys looked longer at the human face, and more boys than girls looked longer at the geometric design. Obviously, a proportion of babies looked equally long at both, but here we're just showing you the data where babies showed a preference towards either the social or the non-social world. So this suggests that whatever the role of culture later on in social experience, there may be a prenatal biological difference that contributes to typical sex differences in the brain and cognition. And if we just extend that last finding to autistic children, this was a study conducted by Karen Pierce at the University of California in San Diego, who looked at autistic kids at the earliest age you could assess them, which is about 2 years old, coming to the clinic. Again, she showed them either a social or a non-social stimulus. What she found was that if children looked for more than 70% of the time at the non-social stimulus, the likelihood that that child was autistic was 100%. So that was an impressive finding when she first published it. Naturally, that was a clinic sample, so we don't know whether you'd get such high levels of accuracy if it was administered in the general population, but looked at side by side with the previous data where we know there's a typical sex difference in social versus non-social interest. What we can see here is that autistic toddlers are showing an extreme of the typical male pattern of interests. So let's move to the final bit of the talk. In the last 5 or 10 minutes, looking at Dervila's face, maybe 5 minutes, we'll talk about prenatal sex steroids, because what we know is that prenatally, there's a surge in the production of testosterone in boys, shown in blue here, in the end of the second trimester and into the third trimester of pregnancy. There's another surge just after birth and a third one during puberty. But prenatally, this is thought to masculinize the body and the brain. And by coincidence, this is also the time when women who choose to have amniocentesis have amniocentesis. So this gives us a little opportunity to measure hormones prenatally, wait for the child to grow up, and look to see if there's any relationship between level of testosterone prenatally and the child's brain and behavior. In our studies, we've focused particularly on a set of prenatal sex steroids called the delta-4 pathway. It starts with progesterone, which gets converted to 17-alpha hydroxyprogesterone, and then androstenedione, and then into testosterone. Each of these precursor hormones, androgens, are converted through the action of an enzyme, and testosterone then is converted to estrogen through the action of aromatase. So we've looked particularly at that pathway, the androgens and the estrogens, looking at these hormones in the amniotic fluid that surrounds the baby during pregnancy. This is all based on a concept developed by Norman Geschwind, who was a neurologist working in Boston in the 1980s, who proposed that these prenatal sex steroid hormones have organizational effects on the brain, which is another word for permanent effects on the brain. And Joe Herbert, who I can also see here in the audience, did a lot of the work looking at hormonal effects on brain structure and function and impact on later behavior. So Mike Lombardo's paper, published in the Journal of Neuroscience now 10 years ago, looked at this cohort of kids and found prenatal testosterone was correlated with brain volume in a number of different brain regions in grey matter. And I'll just pick out some of them for interest: the planum temporale, which is a language area, and the right temporal parietal junction, and superior temporal sulcus, which is thought of as a social brain region, particularly to do with empathy and theory of mind. So this was the first demonstration that in humans that prenatal sex steroids were affecting brain development. We'd known about it for a long time from animal work. In that same cohort, whose mothers had had amniocentesis, we could wait till the child was 4 years old, ask the mother to use the child AQ, or autism spectrum quotient, to look at autistic traits and look for any correlation with prenatal testosterone. And what you see is a positive correlation. So the higher the child's prenatal testosterone, the more autistic traits the child is showing at 4 years old. But in a large study that Mike was a collaborator on, we also worked with the Danish Biobank, because they had big data where amniotic fluid had been stored. So 20,000 pregnancies born in the 1990s. We could link this to the Danish Psychiatric Register to find out who had autism, and we were able to identify 128 who had a diagnosis of autism, and as many controls as we wanted. We selected 219. That mini graph up there shows you, according to when they had their amniocentesis, if you look at the measure of testosterone and other prenatal sex steroids, there is that surge in production just around 14 to 16 weeks of pregnancy. And the result that we found was that autistic children, these were boys, we didn't have enough girls to study, had higher levels of prenatal sex steroids across that delta-4 pathway compared to typical males, who in turn are known to have higher levels than typical females. So at this prenatal hormonal level, we're seeing autism showing an extreme of the typical male profile. And in 2019, we were able to extend this to the study of estrogens, not just androgens, finding that estrogens actually play the bigger role in terms of the odds ratios compared to the androgens down here. So in this kind of cartoon, we can see how testosterone and estrogen are thought to affect the brain. So testosterone can cross into the cytoplasm of the cell. I mentioned that under the influence of aromatase, the enzyme, testosterone gets converted to estrogen. It can bind to an estrogen receptor and enter the nucleus of the cell, where it can act as a transcription factor, changing gene expression, which is thought in turn affects apoptosis, or selective cell death, or pruning in the brain. So the higher your estrogen or androgens, potentially the less apoptosis, which could result in larger brains, more neurons, more neural connections. That's just a kind of model. And this sort of fits with old animal work in the mouse, where if you look at this region of the brain, the sexually dimorphic nucleus of the preoptic area, in a female mouse, it looks a bit like this, but in a female mouse treated with extra estrogens, you see a big change in the neuronal density after administration of the hormone, resembling much more the typical male mouse's brain in this region. So back then, these are kind of classic studies done in animals, but Madeline Lancaster here in Cambridge is now doing similar work in organoids, so that no animals have to be sacrificed, but demonstrating exactly the same findings, namely that the administration of estrogens, for example, changes brain development in humans. A final clue for this prenatal sex steroid theory in autism is looking at women who have polycystic ovary syndrome, PCOS. It's now been shown in 4 different countries. We conducted the UK study, but also in Finland, Israel, and Sweden, that women who have polycystic ovary syndrome, which is associated with elevated levels of prenatal testosterone, are more likely to have an autistic child. So the mother may have elevated testosterone or androgens herself, and this may be crossing the placenta so that the baby is exposed to higher levels of the hormone, or it could be that for genetic reasons, the baby shares some of his or her mother's genes, which may be predisposing the child to produce more of these prenatal sex steroids. And this final clue comes from a study in China, which looked at mothers of autistic children just taking blood samples in pregnancy compared to their child later was diagnosed with autism versus mothers of typically developing children, showing that the mothers have higher levels of testosterone. So suggesting that one source of the prenatal sex steroid elevation in autism may be maternal, but that needs much more research. So just to wrap up, I've tried to both give you the background that genetic factors clearly play an important role in autism, but what we're now learning, but particularly thanks to research in Cambridge, is that prenatal sex steroids are part of the puzzle, that these are clearly interacting and affecting brain development prenatally. And what has still to be understood in much better detail is how this interaction between hormones and genes affects boys and girls who are autistic differently. I'm going to finish there. Thank the many people in the team who are in Cambridge and more beyond who've left. Thank our funders, invite you to visit our website for more information about some of these studies, and we can stop for questions and discussion.
Well, that's it for this series. Thank you for joining us. Thank you, Simon, for a wonderful talk. Catch up on this seminar and all those you've missed on our YouTube channel, and join us back again in October when we'll be launching a whole new series. For more info on the talks covered in this seminar series and all things neuro related in Cambridge, please follow us on Twitter @CamNeuro and follow the links below. See you next time.